So we'll start 17, I guess. Is that okay?
Yeah.
So 17 is fatty acid catabolism. So we're going to talk about how fats are digested in animals, how they're mobilized and transported in tissues, how fats are oxidized, and then how ketone bodies are produced. So let's get on with it. This is a shorter lecture. So.
Was the test Chris?
Yeah. Okay. So, oxidation of fatty acids is a major energy source in many animals. Why is that? Do you guys know? Yeah, it's heavily reduced carbon, so you can do a lot of oxidations and get a lot of energy out of it.
It's hydrophobic, so it tends to stay by itself.
Yeah, it's hydrophobic, so it'll be secluded, from the aqueous part of the cell. So. And then you can store it that way too, so it packs well together when it's all hydrophobic. Yeah. You don't need all that water to go along with it. Like glycogen, when it's hydrated, takes a lot of volume, whereas lipids, they all are sequestered all on their own, right? So about 80% of the energy for our hearts and liver will come from the oxidation of fatty acids. And then also many hibernating animals just rely almost exclusively on fats as their source of energy. And then there's a picture of a grizzly bear. Aw. So fats provide efficient fuel storage. And this is what we were just talking about, right? They're more reduced, not totally reduced. What kind of fat isn't totally reduced? Unsaturated fats, right? So fatty acids carry less water because they're nonpolar. Glucose and glycogen are short-term energy needs, quick delivery. It takes a little more time to get energy out of fat. Fats are for longer term, months and longer. That's why we build fat over time as we get older. So we store it for lean times, right? Which these days, we don't really get too many lean times in modern society. Unless we're broke. But then, typically there's people that'll give us food if we don't, not afraid to ask. So this is adipose tissue, right? Adipose tissue is the tissue in your body that's just to store fat, and these are just fat globules in the adipose tissue. White adipose tissue. What's the other type of adipose tissue?
Brown.
Yeah, and what's the purpose of that?
Short bursts of energy.
Short bursts of energy, but what else? Like for babies, they have it in brown fat's often thought of as babies. What's the difference between brown and white adipose tissue?
A brown adipose tissue?
Yeah, brown's brown, but what makes it brown? It's got a lot more mitochondria, right? So this is burning that fat, and it produces heat also. So I think you can see. I don't know how they stain these. If these are probably just the nuclei. But brown fat is going to have a lot more mitochondria, which will, have iron and stuff in there, making it brown. Or myoglobin also makes it brown, right? So dietary fats are absorbed in the vertebrae, small intestine. So when you eat fat, what happens to it? Can anybody tell me? Didn't you guys go over this in anatomy if you took anatomy? Maybe even just...
This is all bile from the gallbladder.
No, you're reading it right. Yeah, so bile salts are secreted from the gallbladder, and they're there to emulsify the lipids that you've eaten. So they emulsify it, and that also puts them in contact with what?
Emulsification?
Yeah. That makes them available for those lipases. Digestive lipases, which will break them down into single free fatty acids and mono and diacylglycerols, and then they'll be absorbed by your intestine cells. And then your intestines, your epithelial cells, will then put them back together as triglycerides and cholesterol esters, and package them into chylomicrons, right? Do you guys know what chylomicrons are? They're a lipoprotein, so they're a lipoprotein particle that transports fats from the intestine to your liver through your yeah. And what happens, you break them down to absorb them, and then they get packaged into these chylomicrons with certain marker proteins, such as ApoC2. You make a chylomicron, then it's in the circulation. As it's in the circulation, there's lipoprotein lipases will break off free fatty acids, or break free fatty acids down from the triacylglycerols, and as it circulates, it gets more and more dense, meaning less and less lipid there, right? So there's less and less lipid, more cholesterol, and more protein content. So it's less or more and more dense, and then cells in the circulation will absorb the free fatty acids in it as energy. And then it ultimately will go to your liver and your adipose tissue and be stored, right? So. And this is an image of a chylomicron. So you have cholesterol esters and triacylglycerols, and then phospholipids along the outside, right? That's going to interact with the aqueous phase of the plasma or the lymph. So hydrolysis of fat yields fatty acids and glycerol, right? You guys remember that? So triacylglycerols has one backbone molecule that has 3 carbons. Oh, I always pick this bad marker. That's bad. Right? So triacylglycerols are ester bonds with 3 fatty acids with 1 glycerol. So when you hydrolyze that, you end up with fatty acids, which are, the long, well, they don't necessarily have to be long. Fatty acids plus triacylglycerol, right? And then some lipases are regulated by hormones, right? So if you remember epinephrine, that means we need energy now, fight or flight. We need energy to survive. Glucagon means just our body's out of glucose. Blood glucose levels are low. We need to start kicking in glucose again. And it can also trigger kicking out, starting to burn down lipids to produce glucose, and also starting to produce ketone bodies. So ketone bodies are the substitute for sugars in our blood when we're really low in sugar. And that's because, anybody know why we use ketone bodies?
Well, if you're out of food, just ketones are out of food.
Why can't we just send out the free fatty acids into the bloodstream?
But there's no free fatty acids to get.
No, but you make ketone bodies from the fatty acids.
I'd say it's an intermediate between the burning fatty acids and actually using fatty acids. So you get that 2 sources of energy.
Well, the ketone bodies, they're there because they don't need any sort of transport molecule. Okay? So ketone bodies, they're soluble in water, like acetone.
Right.
And they'll just mix with the water and circulate in your bloodstream like glucose normally would, just free. Whereas fatty acids, they have this big hydrophobic tail. They're not very soluble in water. I mean, they are because they have the head group, but they don't freely transport around in the bloodstream. They're absorbed to a protein. They're bound to a protein in the bloodstream. Anybody know what protein that is? That binds and transports free fatty acids?
Isn't it the HDL and the stuff, or that?
No, those are diacylglycerols, triacylglycerols, and cholesterol esters of glycerol. But free fatty acids are actually transported around with albumin. So albumin will bind several fatty acid molecules, and that's how it gets transported around. So that's why we make ketone bodies, but that's at the end of the chapter. So hormone triggers the mobilization of stored triacylglycerols. So glucagon, what does that tell us? Low blood glucose. So it's telling us we need more blood or more glucose in our blood. activates adenylyl cyclase, which activates protein kinase A, which will phosphorylate hormone-sensitive lipase, causing it to be active. And it will break down the triacylglycerols to diacylglycerols to monoacylglycerols, and you'll release free fatty acids, and they'll secrete it in the bloodstream, or they'll get picked up. You see this? Serum albumin. Serum albumin transports them, and it'll take it to the liver, and they can actually absorb the free fatty acids once they're bound to the bloodstream. So, bound to the albumin. So, fatty acids and glycerol. So this shows us, what are the difference between these two?
Just the double ones.
Yeah, one is a saturated fatty acid, one's an unsaturated fatty acid. And it's a cis unsaturated, which is typically what we get in our diet, unless we're eating trans fats, which is where you take poly or monounsaturated fatty acids, you hydrogenate them, or partially hydrogenate, and they'll flip around to the trans when you excite them like that. So glycerol from fats can actually enter glycolysis, right? So fats are triacylglycerols or diacylglycerols are di or triesters of free fatty acids. The fatty acid part cannot be made into sugar, whereas the glycerol can, right? And this is how that glycerol will enter glycolysis. So you have glycerol, you have to activate it with ATP to form glycerol 3-phosphate by glycerol kinase. So that's an enzyme that's going to add phosphate to glycerol. Then you have to oxidize it. You oxidize it with glycerol 3-phosphate dehydrogenase, and the NAD will pick up that hydrogen, take those electrons away. And then you have dihydroxyacetone phosphate. And does that sound familiar from glycolysis? Dihydroxyacetone phosphate.
It does, yeah.
Yeah, and triose phosphate isomerase will convert that into D-glyceraldehyde 3-phosphate. So that's how you can get sugar from glycerol. Remember, you can't get sugar from the free fatty acids, but glycerol is the only part of fat that you can make sugar out of. Glycerol is activated by phosphorylation. Now, these are just the steps that we just went through, right? So glycerol kinase phosphorylates. So you can't just add the phosphate, you need ATP to do that. And then it's oxidized with NAD, accepting those electrons to dihydroxyacetone phosphate. And then triose phosphate isomerase will then convert that to D-glyceraldehyde 3-phosphate. And then it'll go on through like normal glycolytic pathways. So transport or attachment to phospholipids requires conversion to fatty acyl-CoA. So once you get that free fatty acid into the cell, it has to be converted to fatty acyl-CoA to move around the cytosol. Unless it's a light chain free fatty acid. So and this occurs this way. First you activate it by adding adenylate. Basically it's called adenylation, where you add the AMP portion of ATP to the free fatty acid. So you have this, what kind of bond is this between two acids? Do you guys remember?
Covalent.
Well, yeah, it's covalent.
That's
But you know, like between an acid and an alcohol is an ester. Between two acids is a, huh? Not enol, huh? Anhydride, right? Yeah. So this is a organic phosphoanhydride, and then an adenosine, which is the A part of your RNA or DNA. So then coenzyme A will come in and attack that carbonyl on the carboxylic acid group, and then you have fatty acyl-CoA, which is the same thing as acetyl-CoA, but it's a much longer carbon chain, right? Acetyl-CoA is the smallest fatty acid you'll have, probably. I've never heard of formic acid-CoA or formyl-CoA. Have you? No. But so I've never heard of that. It could exist, I guess, because I claim to know everything, and I don't know much. Okay, so coenzyme A will attack that acyl adenylated acyl, fatty acyl adenylate rather, and then you'll end up with the acyl-CoA, and that's done by fatty acyl-CoA synthetase, and you get AMP released as a product. And also, when you form that fatty acyl-CoA molecule, you get pyrophosphate, and that's kept at really low concentrations in the cell also, because it's broken down by inorganic phosphatases to release 2-phosphates. So this really low concentrations, it goes to 2-phosphates really fast, and then you get energy out of this. So you get a lot of energy out of that process to form that acyl-CoA. But you're using an ATP to do it, right? So fatty acid transport in the mitochondria. So these acyl-CoA molecules can't cross the mitochondrial membrane. So they can diffuse, they can be transported to the mitochondrial membrane, but there's a transport system to get them into the mitochondria. And that's where beta oxidation takes place, where you're extracting energy from these fatty acids. So fats, they're degraded into fatty acids and glycerol in the cytoplasm. Fatty acids are transported to other tissues for fuel. Beta oxidation occurs in the mitochondria. Small fatty acids can diffuse freely, but not the larger ones. Anything above 12, it has to be transported in the mitochondria. And it does this with this acyl-carnitine transporter that's at the mitochondrial membrane. And this is picture carnitine. You have this quaternary nitrogen, so it's positively charged, and then the carboxylic acid group on the other end, negatively. So overall, it's got a net zero charge. It makes, in this state, it's a, what's it called? Oh, never mind. It's carnitine. Huh? Polar. Well, yeah, it's polar, but it's like amino acids that are in solution in water. They're, I forget the word. Huh? No, where it carries 2 charges of opposite, so it's got a net zero charge. What's that word? I can't think of it now either.
Oh, is it this, not this Twitter or something?
Yeah, zwitterion, exactly. That's the word I was looking for.
Ask me that on a test.
Okay, and this is the carnitine transporter. So you have out here in the cytosol, coenzyme A will go to the carnitine transporter, where we'll substitute the coenzyme A for carnitine, and you'll have an acylcarnitine formed at the membrane. It'll be transported by the carnitine acyltransferase 1 through the intermembrane space to another transporter, where we'll take off the carnitine, which will go back and form coenzyme A again in the matrix of the mitochondria. So that's carnitine acyltransferase 2 that removes the carnitine and sends it back the other way. So remember that fatty acids have to be covalently bound to carnitine to get into the mitochondria for beta oxidation. And this is another picture showing you the same thing. So coenzyme A, carnitine comes off, which hydrolyzes the sulfhydryl coenzyme group, and then you form carnitine. Acylcarnitine gets transported into the mitochondria, where the process is reversed. So
is carnitine transported in via ATP, or is it simple diffusion?
no, typically it'll go down. It's
Downgrading.
yeah, because you're gonna get rid of that acyl fatty acid once you're in there, if you're low in energetic state. So stages of fatty acid oxidation. So we're going to burn these fats now that they're in the mitochondria. The first stage consists, we're going to oxidatively convert that big, long, free fatty acid into 2 carbon units. What are the 2 carbon units called?
Acetyl.
Yeah, acetyl. And there'll be acetyl-CoA groups too. So we're going to convert like a big, long, free fatty acid.
They're always even?
They're not always even.
I read something, it was in the focus? Like they're mostly...
They're mostly even. Yeah, and then you end up with 2. And we're going to talk about when they're odd, we end up with an odd product, propanoic acid, or propanol-CoA, right? And then that gets converted, I think, into something else that we'll talk about.
Isn't there only one carbon in formic acid?
Yeah, there's only one carbon in formic acid. But yeah, I've never. We stop at propionic acid, and then that gets converted into an intermediate in the TCA cycle, succinate, I think. But I'll remember when I see it on the PowerPoint. Okay. And then, so when we do that, we're going to generate a lot of NADH and FADH2, which we'll be able to extract those electrons from in the what do you call it? Electron transport chain. Yeah. And this involves oxidation of the beta carbon to the thioester of the fatty acyl-CoA. Stage 2 involves oxidation of acetyl-CoA into CO2 via the citric acid cycle, so we'll get a lot more NADH and FADH2. And then stage 3, we're just going to generate the ATP from all the NADH and FADH that we generated by burning those fats. Okay, and this is the picture. We have a big, long, free fatty acid. You know, that'll come into the cell. We'll make acyl-CoA, so it'll be a coenzyme A group. It'll diffuse to the mitochondrial membrane where it'll be transported via carnitine. So carnitine acyl, or acyl-carnitine will be transported in, then converted back to acyl-CoA, where we'll oxidize it to 2 carbon units. Those 2 carbon units will enter the citric acid cycle like normal, and we'll get all the NADH and FADH2 and make a lot of ATP via the electron transport chain. So the beta oxidation pathway, so each pass removes 1 acetyl moiety in the form of acetyl-CoA. So again, coenzyme A is involved, and it involves basically 4 reactions. Acyl-CoA dehydrogenase. So if it's a dehydrogenase, what do you think that's the first reaction is going to do?
It's FAD to FADH.
Yeah, so what's it doing? What's happening to that fat? Yeah, you're removing a hydrogen, so that's an oxidation, right? Because you're oxidizing it. So we take, if it's a saturated free fatty acid, or if it's a saturated fatty acid, we convert it from saturated fatty acid to an unsaturated fatty acid. We introduce a trans delta 2 ene group, enoyl-CoA. So this is a trans fatty acid that we produce in our body during metabolism. So then we hydrate that using enoyl-CoA hydratase. So we just add water across this double bond, and we end up with an alcohol, L-beta-hydroxyacyl-CoA. So then we get one more oxidation. See a dehydrogenase, it's an oxidizing step. Beta-hydroxyacyl-CoA dehydrogenase. And these enzymes, they tell you they're substrates pretty much in the order they're going. So dehydrogenase, this time NAD accepts the hydrogen, and we produce a ketone, beta-keto-acyl-CoA. And then coenzyme A will come in, attack this ketone, and cleave it. It's acyl transferase thiolase, so it's a thiolase reaction, which removes the first acetyl-CoA group, and then you're left with an acyl-CoA that's 2 carbons shorter than you started with. And this will go on and do all those 4 again.
Until it gets down to the second.
7 more times until you're down to the final acetyl group. Or if it's an odd one, you'll go to a propionic group. So step 1 is the dehydrogenation of alkane to an alkene. Remember, alkanes are saturated. Alkenes have an ene group, the double bond. Catalyzed by isoforms of acyl-CoA dehydrogenase on the inner mitochondrial membrane. So there's 3 different isochains. One's for very long fatty acids, 12 to 18, medium chain fatty acids, 4 to 14, or short chain fatty acids, 4 to 8. Why don't we care about 2 or 3 length fatty acids?
Well, because they just...
That's the end product of this whole thing, right? So, yeah.
Isn't there resonance frequency between the 2 and the 3.
Resonance what?
The resonance harmonization between the 2 and the 3 bonds.
What 2 and 3 bonds?
At the end of the chain, there's a resonance harmonization that you find.
You mean like with, you have the carboxylic acid group?
Yes.
That's in a thioester group? A thio, right? That's a thioester.
There's a carbon, 2 carbon, and then 3 carbon that share double bonds.
Oh, that share double bonds, like that, yeah. So this can come up here, right? And then this will come up here. And then, right? Is that what you're talking about?
That's what I was thinking about. I might have tried that.
And then this will give this a positive charge, and this will give this a negative, partial negative charge, yeah. So this step 1, dehydrogenation of alkane to alkene, different enzymes based on the length of that fatty acid, results in a trans double bond, which is different than what we normally eat, a cis double bonds, but this trans double bond's basically an intermediate in burning of the fatty acids. It's analogous to the succinate dehydrogenase reaction in the citric acid cycle, right? So electrons from the bound FAD transfer directly into the electron transport chain, just like that succinate dehydrogenase reaction in that. So what happens to the succinate? Is it fumarate? Yeah, where it's got the double bond, right? And then fumarase adds the water across it, right? Yeah, so it's basically the same thing, but this time we're coenzyme A bound fatty acid that we do this to. And this is the overall reaction. Oh, this is the step 2, adding the group, the water. So catalyzed by 2 isoforms, enoyl-CoA hydratases. So hydratase, you're just adding water to a double bond. Soluble short chain hydrotases is chronotase. Membrane bound long chain hydratases are part of the trifunctional complex, and water adds across the double bond yielding an alcohol. It's analogous to the fumarase reaction in the citric acid cycle. So you get the same stereospecificity. So here you have your trans-enoyl-CoA chronotase, which is soluble short chain. So this would be a short chain. Water just comes in and you form an alcohol across that double bond, right? So, very stereospecific too. So these are the first 2 steps. You have your saturated fatty acid. Oxidize this beta carbon. Now you have your ene group, and then water can come in and form the alcohol. The next step's going to be, this is the FAD cofactor, right? So this is what does the first oxidation. So we go from oxidized FAD to the reduced FAD. We get 2 electrons and 2 hydrogen ions total that form this. So dehydrogenation of the alcohol, it's catalyzed by beta-hydroxy-acyl-CoA dehydrogenase. It uses NAD as a cofactor, and basically we just pick off the hydrogen again with the NAD. The electrons here in the alcohol will drop back down, and then you'll form a beta ketone. So your alpha and then beta ketone. Beta-keto-acyl-CoA. And then the final step is catalyzed by acyl-CoA acetyltransferase, also known as thiolase. So thiolase, what do you think of when you hear thio?
Sulfur.
Yeah, sulfur. Thiolase, you use acetyl-CoA, or not acetyl-CoA, but coenzyme A, the thiol group, to do that splitting of the molecule, hence thiolase. So it's breaking it apart, right? The carbonyl carbon of the beta-keto-acyl-CoA is electrophilic, and the active site thiolate acts as a nucleophile and releases the acetyl-CoA. Terminal sulfur then on coenzyme A acts as a nucleophile and picks up the fatty acid chain from the enzyme. So it's actually a cysteine group on the enzyme that you form a covalent intermediate with during thiolase. The net reaction is thiolysis of a carbon-carbon bond. And this is the overall mechanism for this, right? So you have an enzyme with a cysteine thiol group, it attacks that beta keto group. So these electrons, they pop up, and then you form, this is a quaternary intermediate. The electrons will drop back down. These electrons will then go, and you've cleaved this bond. You have a carbanion intermediate that will pick up a hydrogen off a base on the enzyme, and then you release your, acetyl-CoA. And then the next step is here. You still have this covalently bound 2 carbon shorter free fatty acid. Coenzyme A then comes in and attacks that carbonyl, and you get release of the enzyme and acetyl-CoA that's 2 carbons shorter than it was before. And this is the overall steps 3 and 4 here. 2 carbons shorter. So the enzyme that does this it's a trifunctional protein. So it has 4 alpha subunits that contain the enyl-CoA hydratase activity. That's, if you remember, the hydratase adds the water across the double bond. Beta hydroxyacetyl-CoA dehydrogenase activity. So that what's the, do you guys remember? I just told you a second ago, so I'm not expecting you to. What does the oxidation here? What's the electron acceptor for this reaction? No, the NAD. So it's a dehydrogenase. We're removing a hydrogen. NAD does this. It's responsible for binding to the membrane also. Then there's 4 beta subunits that has the long chain thiolase activity. That's where you get that sulfhydryl group on a cysteine that does the attacking. May allow substrate channeling, and that's because those enzymes are close together. Channeling just means one product goes immediately to the next functional part of the enzyme and so forth. So you don't have a buildup of any products or diffusion of products away to form, other products. It's associated with the inner mitochondrial membrane, processes fatty acid chains with 12 or more carbons. Shorter chains are processed by soluble enzymes in the matrix itself. So for palmitidic acid, repeating the 4 steps 6 more times, you get 7 steps that go through that before you end up with, acetyl-CoA left. FAD is formed in each cycle, so that's 7 total. NADH is formed with each cycle, so 7 FADH2s and 7 NADHs. And then acetyl-CoA enters the citric acid cycle and further oxidizes into carbon dioxide. And this makes more GTP, NADH, and FADH2. And then electrons from all of the FADH2 and NADH are entering the electron transport chain. So each round produces an acetyl-CoA and shortens the chain by 2 carbons. So beta oxidation, pretty simple. And then NADH and FADH serve as a source of ATP if they go through the electron transport chain. There's one place in a cell, especially plant cells, where the NADH or FADH2 doesn't do this. Anybody know where that is? You ever heard of the peroxisomes?
Peroxisome attacks the same way as the mitochondria, right?
Yeah, but there's no electron transport, so you lose all of that energy that you would normally have through that you would have normally formed through the FADH2 through the electron transport chain. In peroxisomes, those electrons go from FADH2 directly to oxygen. And that's going to release heat, and then you form hydrogen peroxide when you take take the electrons from FADH2, add it to oxygen, you end up with hydrogen peroxide. And then hydrogen peroxide gets broken down into water
and oxygen.
and oxygen again via catalase. So that's not this step. So the mechanisms of beta oxidation, they're similar mechanisms as in other metabolic pathways, and we already talked about the citric acid cycle, that's similar. And then it's also similar with the oxidation of isoleucine, leucine and valine. So oxidation of unsaturated, all of that was just for saturated fatty acids. When you introduce a cis double bond somewhere in that fatty acid chain, it's going to change things up just a little bit. So naturally occurring unsaturated fatty acids contain the cis double bonds, and they're not a substrate with the enoyl-CoA hydratase, because that takes that trans double bond, right? 2 additional enzymes are required. Isomerase, which basically converts the cis double bond starting at carbons 3 to a trans double bond. Or reductase also reduces the cis double bond that's not at carbon 3 to saturate it, gets rid of that. Okay? Monounsaturated fatty acids only require the isomerase. Polyunsaturated fatty acids are going to require both. And we'll see in a second how that works. So for monounsaturated fatty acid, here you have double bond on carbon 9. Normal beta oxidation will take place for the first 3 cycles, and you release 3 acetyl-CoAs. But then now you're here to this, what's this, alpha, beta, this cis delta 3 dodecanoyl-CoA group. So it's a cis delta 3 bond, so you have the ene group on the number 3 carbon. And then delta 3, delta 2 enoyl-CoA isomerase isomerizes that, switches the double bond to here and converts it to trans delta 2 dodecanoyl-CoA. So you take the cis delta 3 and it converts us to to trans delta 2. And then this is a substrate for normal beta oxidation.
This was a question I had earlier about the harmonization between the cis and the trans.
Yeah.
I was wondering if that's replicated in the other chemicals, like the one you had on board,
If it's
COH, COA.
Oh, like if you do this.
Yeah, the acetyl COA and that.
So what's going on?
Right, the 2 and the 3 bond on the acetyl COA. I thought that they harmonized.
So this is 1,
and
2.
What we have at the 6 acetyl COA.
Oh. Like this, right?
Yes.
Oh, so you're like if this will come up.
That goes back and forth to provide
Yeah, this will come up,
oxygen and
leaving this right here. How does it leave this? So if this comes up, this carbon will be partially positive, right? There's going to need for electrons to go here, which this one won't be able to provide unless it goes from here and does that. But then you need something to give electrons here. So there's really no resonance with this structure and what we had before.
okay.
So that's why you need the enzyme to get that, to do that. Okay? So polyunsaturated acids needs that isomerase and the reductase. And again, for this example, linaloyl-CoA, where it has the cis delta 9, cis delta 12. First 3 cycles are normal beta oxidation. You'll release 3 acetyl-CoAs. And then you have the delta 3, delta 2 enoyl-CoA isomerase, which converts this again to a trans delta 2. And then one more cycle of regular beta oxidation, you release an acetyl-CoA. And then you have, this is the result of that normal oxidation. You get your first oxidation in the second cycle. You end up with this trans, and then you have a cis here. So this has to be reduced then and converted. So you use NADPH for those reducing cofactors to form NAD. The electrons come from NADPH, and then you make that into a saturated bond, basically. And then it can continue like normal. normal beta oxidation after that. So you end up with trans delta 3, and then that enoyl-CoA isomerase converts it to trans delta 2.
Delta 3, delta 2 enoyl-CoA isomerase, is that specific to only double bonds in the delta 3
Yeah.
2 position?
Yeah.
Okay.
Number 2 carbon and number 3 carbon position. So first double bond requires isomerization. So this is what we just looked at. So we isomerize the first double bond. Beta oxidation will continue for one cycle, and the first oxidation of the second cycle, where you'll form this trans delta 2 cis delta 4. Okay, and then the 2,4-dienoyl-CoA reductase just reduces that, and you end up with the trans delta 3. Enoyl-CoA isomerase will convert that to trans delta 2, and then it'll continue normal beta oxidation after that. So now we were talking about odd fatty acids. So for odd numbered fatty acids, it's not the most abundant kind of fat that we're eating. Most dietary fats are even. Many plants do synthesize odd number fatty acids. And the final product we get from this is propionyl-CoA forms as the last beta oxidation step. Bacterial metabolism in the rumen of ruminants also produces propionyl-CoA. So in cows, they'll produce that. I think we'll probably produce some of that also in our gut from bacteria, which is supposed to be good for like cholesterol and heart disease. But anyways, propionyl-CoA carboxylase will come in, and what does a carboxylase do? Yeah, adds a carboxyl group to it. So propionyl-CoA carboxylase adds a carboxyl group, which converts the propionyl-CoA to methylmalonyl-CoA, which is this compound here. Methylmalonyl-CoA epimerase then just rearranges that and produces L-methylmalonyl-CoA, which then methylmalonyl-CoA mutase converts the L-methylmalonyl-CoA to succinyl-CoA. And then succinyl-CoA, does anybody remember what do we do with that?
It's a block of the citric acid.
Yeah, so it takes succinyl-CoA, goes on and does that, what's it called, substrate level phosphorylation to form, a nucleotide triphosphate in that part of the citric acid cycle, and then it'll continue like normal through the citric acid cycle. So first step is adding that carboxyl group. So propionyl-CoA carboxylase takes bicarbonate and ATP. Does anybody remember that? That always uses biotin. It's that coenzyme that's on a chain, basically. It'll go from one subunit to the other. It'll pick up the carboxyl group in ATP. ATP phosphorylates the carboxyl group to form carboxyphosphate. Then the biotin will pick up that carboxyl group after that and then transfer it to the propionyl-CoA to form D-methylmalonyl-CoA. Then it just gets rearranged. D-methylmalonyl-CoA via methylmalonyl-CoA epimerase forms L-methylmalonyl-CoA. And then methylmalonyl-CoA mutase converts that to succinyl-CoA by another rearrangement. And then directly into the citric acid cycle. Now, methylmalonyl-CoA mutase uses coenzyme B12. Anybody ever heard of that? Cobalamin?
Yeah.
Yeah.
There's a big article on it right in the book.
Oh yeah?
Yeah, it shows you the structure, how it's nucleotide and the transformed nucleotide on the bottom. It's got a cobalt right in the middle.
Yeah.
Got a nice little structure.
Does anybody know what's interesting about the mechanisms cobalamin uses typically? So it'll be one of the places in organic chemistry where you, instead of using the arrows, you'll use a half an arrow. What's that when only one electron goes and the other electron will go somewhere else? Yeah, so it has radical chemistry. So isomerization requires cobalamin or coenzyme B12, like we said. And this is the overall structure of cobalamin, or B12. So yeah, you have your handles there on either side, deoxyadenosine, and then adenosine down here, or dimethylbenzimidazole ribonucleotide, rather, which coordinates the cobalt, and then you get that interesting chemistry from that corrin ring system. So regulation of fatty acid synthesis and breakdown. So again, it's regulated through hormones, right? So high blood glucose, you secrete insulin. Insulin tells you to store energy, right? Not burn energy. So it basically, through several steps, it'll block transport of acetyl-CoAs into the mitochondria, so you won't be able to burn them. And it does that, so insulin activates phosphatase, which then removes the phosphate from acetyl-CoA carboxylase. That's what ACC stands for. I don't know if they're introducing you to this a few chapters early, because acetyl-CoA carboxylase is the enzyme that forms malonyl-CoA from acetyl-CoA when you're producing or storing fatty acids. So then insulin will trigger any of the acetyl-CoA around to be produced into a new fatty acid. It's fatty acid synthesis. Whereas protein kinase A, in response to glucagon, will phosphorylate acetyl-CoA carboxylase, blocking synthesis of new new fatty acids, and allowing fatty acids to be transported into the mitochondrial matrix for beta oxidation to occur. So like I was saying, in the mitochondria, you can extract all the energy from your cofactors, those electrons. They'll transfer that energy to the electron transport chain, where you'll be able to produce that hydrogen gradient across the inner membrane and produce ATP via oxidative phosphorylation. The peroxisome, however, will take those electrons and transfer them directly to molecular oxygen and produce energy released as heat. And then hydrogen peroxide is eliminated by catalase after that. And this just shows you the two. So the first step, we take a saturated fatty acid chain and we oxidize it with FAD to produce an ene group, and that FADH2 in the peroxisome will go give its electrons directly to oxygen and form hydrogen peroxide. So, and then it'll go on like normal, and then NAD in the peroxisome will be exported for reoxidation. And then you'll produce the acetyl-CoA at the final product is also exported out of the peroxisome. Whereas if you're in the mitochondria, it would go to the TCA cycle. So the acetyl-CoA in the glyoxysome is then converted to a metabolic intermediate. Remember, plants and bacteria can take fats such as acetyl-CoA and convert it to compounds that can be converted to sugar. So plants can convert fats to sugar, and so can bacteria, but not mammals or other animals like us. So this is just fats. I mean, just plants, right? So formation of ketone bodies. So ketone bodies, as I was saying at the beginning of the class, they're a transportable form of the fatty acids where you don't need any carrier protein. Normally you need albumin to carry around free fatty acids, but we form ketone bodies because they will dissolve in your plasma and be transported around your body just like sugar would. So, and we've three kind of ketone bodies that we find. So hydroxybutyrate, acetoacetate, and what's the last one? Acetone. So entry of acetyl-CoA into the citric acid cycle requires oxaloacetate oxaloacetate. When oxaloacetate is depleted, acetyl-CoA is converted into ketone bodies, which frees coenzyme A for continued beta oxidation. The first step is the reverse of the last step of beta oxidation, thiolase reaction, which joins two acetate units together. So you take two acetyl-CoAs, a reverse thiolase reaction produces acetyl-CoA, and then HMG-CoA synthase takes another acetyl-CoA, which produces beta-hydroxy-beta-methyl-glycerol-CoA, HMG-CoA. So, and then HMG-CoA lyase will lyse that, releasing one acetyl-CoA, and then you end up with acetoacetate. Acetoacetate can go two different directions to form beta-hydroxybutyrate or acetone. So all three of these can be transported around in your plasma as an energy source. So your brain can use these, but it's not as happy with these as it would be with normal sugar. So this is, this is the goal of those low sugar diets, is to produce these ketone bodies and use your fat tissue to make these ketone bodies to give your body energy that it needs. So then the release of free coenzyme A. So acetyl-CoA, two acetyl-CoAs, the reverse thiolase reaction releases the coenzyme A. Acetyl-CoA via HMG-CoA synthase produces this beta-hydroxy-beta-glycerol-CoA. And then the formation of the ketone bodies. Beta-hydroxy-methyl-glycerol-CoA forms acetoacetate, and that can be decarboxylated to form acetone or reduced to form beta-hydroxybutyrate. So ketone bodies are produced in the liver during starvation and diabetes. So if you have type 1 diabetes, your cells are starved because they can't transport sugar into the cells, so they want ketone bodies, and your liver will start to produce ketone bodies. Ketone bodies are released by the liver into the bloodstream, and organs other than the liver can use ketone bodies as fuels. High levels of acetoacetate and beta-hydroxybutyrate lower blood pH, and that can lead to like diabetic ketoacidosis is one of the reasons, one of the results of this ketogenesis. Okay, so that's why like some diabetics will have that acetone breath when they're undiagnosed. That's one way you can diagnose them, is because they actually have acetone on their breath that they're producing.
Isn't it like a sweet tasting breath?
Sweet tasting breath? Acetone.
That's the stuff that gets rid of nail polish.
Yes.
Yeah, it's horrible. It eats plastic. It ruins your brain.
It's also an energy source for your brain when you don't have sugar.
Don't drink it.
No, don't drink it. Please don't drink acetone. So.
Is it not harmful to our body, or is it at the doses that or the?
Yeah, you're not going to produce it.
Okay.
It is harmful because it'll produce that ketoacidosis, right? But when you need energy,
If it's on your skin, it's like it ruins your skin. So that's why I'm like.
I don't know, I get it on my skin. It doesn't. That's why it's fingernail polish, right?
Yeah.
Because it doesn't ruin your skin. Well, it doesn't burn you.
Okay, maybe I'm just really sensitive. I don't know.
Maybe it dries out your skin.
Yeah.
But it's not going to burn you.
No.
Yeah. So. Like you could also take off fingernail polish with xylene, but that would probably burn your skin.
I don't think I can get that from this.
No, they sell it at Home Depot and Lowe's. It's paint strippers.
Yeah. That's not okay.
So, but xylenes are nasty. But acetone not as bad.
Yeah.
So, and this is just figure showing you hepatocytes converting acetyl-CoA to form ketones in response to low blood sugar. Acetyl-CoA will also they'll take oxaloacetate. If you have low blood sugar, your liver cells will take oxaloacetate to produce blood glucose, which will remove things from, intermediates from the citric acid cycle, and acetyl-CoA will also be sent towards making ketone bodies at the same time. So, and then the acetyl-CoA, the ketone bodies will be transported through your body. So that's the end of chapter 17. Fats are an important energy source for animals. You remember, beta oxidation converts the long chain fats to the 2 carbon units and produce a lot of NADH and FADH2 because fats are so reduced to begin with. Acetyl-CoA formed in the liver can then be oxidized via the citric acid cycle or converted to ketone bodies, and that's going to occur based on how much, if you have lower normal blood sugar, right? And it'll serve as fuels for other tissues. And that's it for today, I guess.